Bioenzyme.
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 Bioenzymes.
Bioenzyme. Bioenzyme is the best.
You can use bioenzyme in your daily life and make your life chemical free and save environment. Bioenzyme.
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 Bioenzyme. Bioenzyme.
Bioenzymes.
Bioenzyme! Bioenzyme is the best!
You can use bioenzyme in your daily life and make your life chemical free and save environment.
I request our speakers, Shri Patanjali Jhaji, Professor Dinesh Mohanji, Dr. C. Arunji, and Dr. Susan Titus Ji to please come up on the stage. I also request Professor S. Garzeji and Professor S. Mazumdarji, who are the chairpersons of this session, to be on the stage, please.
About 12 years back, when we had bought this land, it was two-thirds of it was barren. My parents lived here, and they started doing plantation. And we essentially stuck to natural farming. Through the journey, we learned that it's the trees which give the maximum resources. Tilling of a soil is a crime. And
Good afternoon, everyone. I hope everybody's had their lunch and are now refreshed for our next technical session. So it gives me immense pleasure to welcome all of you to the second technical session on bioenzymes and agriculture. This seminar is a part of G20 Summit on Environment, Sustainability and Bioenzymes, taking place on the occasion of Earth Day celebrations on 21st April 2023 at the University of Mumbai. Organized by the Department of Biotechnology and Academy of Administrative Careers, University Mumbai, in association with Society for Innovation in Biosciences and Bioenzyme Entrepreneur Academy of India. I'm honored to chair this session along with Professor S. Garze, Head Department of Chemistry, University of Mumbai, and Associate Dean, Science Faculty, University of Mumbai. Professor Garze's research is focused on development of simple, eco-friendly, single-source molecular precursors, SSPs, which can lead to phase-pure and uniform morphology, nanoparticles, and thin films. The SSPs contain desired elements which are required in the final material in a single molecule. They have some distinct advantages over conventional multiple-source precursors. These include their low toxicity, no or limited pre-reactions, control or stoichiometry, control on volatility using suitable ligands, etc. He has remarkably contributed in his area of research by publishing various research articles. Dr. Garze has been awarded with Performance-Based Incentive Award, Boys Cast Fellowship, Young Scientist Research Award, Professor A.K. Day Young Scientist Award, and many more. He has completed many research projects granted by BRNS, DST, and many more, and guided many PhD and MSc research students. Along with Professor Garze, we have Professor S. Majumdar, Senior Professor, Department of Chemical Sciences, Tata Institute of Fundamental Research, Mumbai. Dr. S. Majumdar and his students are involved in the study of structural mechanistics aspects of the biochemical function of various electron transfer and redox metalloenzymes using different physiochemical techniques, as well studies on wild type and specifically designed mutant proteins are being carried out in the laboratory. The metalloenzymes currently being studied are cytochrome C oxidase, cytochrome P450 and preoxidases. He is also involved in study of enzyme kinetics, protein stability, and unfolding refolding processes, protein engineering. The main techniques are used in his studies are circular dichroism, microcalorimetry, protein mass spectrometry, ESIMS, and MALDI-MS, protein engineering techniques, high resolution NMR, and time resolved fluorescence. He is the Secretary of National Academy of Science India, NASI, Mumbai chapter. We are fortunate to have with us today a panel of distinguished speakers who will share their expertise in this field of bioenzyme and agriculture. Our esteemed speakers include Mr. Patanjali Jhaji, IRS, who's the former Principal Chief Commissioner of Income Tax in Mumbai and Director General Investigations for Karnataka and Goa. In his career as a tax administrator, he played instrumental role in unearthing massive tax evasion schemes. Over the last 20 years, he has created over 100 acres of food forest, now named One Near Farm. With a range of trees having commercial and medical value, the forest is self-sustained with pollinating bees and grasslands. The farm has now become a proof of concept of no-till, no external input, and multi-layer farming, and has acted as a source of inspiration for many to raise and switch to natural farming and raising food forests in place of traditional destructive farming practices, leading to coming up of food forests over 1,000 acres of land. He's been dubbed as the hero of sustainability for 2020 by the Better India and also applauded by India Today. He received Vivekananda Sustainability Awards for sustainable agriculture practice with sustained improvements in yields, farmers' income, and improved environmental profile in 2022. He received Parakram Award instituted in the name of Netaji Subhash Chandra Bose in 2023. He's worked in close tandem with the eco tasks of Indian Army in its afforestation efforts. He has worked to make known the effectiveness of the vetiver system and have gifted vetiver slips for the afforestation and landscape stabilization and decontamination efforts of the Eco Task Force in Marathwada, Samba sector, banks of River Ganga, etc. Our second speaker for this session, the second panelist, Dr. Susan Titus, who's a Naval Material Research Laboratory in DRDO, Ambernath, Thane, Maharashtra. Dr. Mrs. Susan Titus is a scientist, G-Head, Marine Biotechnology, Naval Materials Research Laboratory, DRDO, Ministry of Defense, Shil Badlapur Road, Ambernath, Thane. She has 7 patents and 35 papers on her name. She was awarded with the STE Women Excellence Award in 2019, DRDO Laboratory Technology Group Award for Excellence in R&D in 2019, DRDO Laboratory Scientist of the Year Award for the Excellence in Research in 2008, and DRDO Technology Day Commendation Medal in 2004. President's Guide Medal in 1978, a Merit Scholarship awarded in 1978 as well. In research area, her achievements include accelerated bioremediation technology for marine oil spill, process for production of biosurfactant from hydrocarbon utilization bacteria, process of microbial biodegradation of polyethylene, LDPE, bio-based anti-fouling coatings for control of marine biofouling, and fiber optic biosensor for detection of waterborne pathogens. Her present research is on development of bio-clean solution for oily surfaces and green synthesis of butane trial by biotech route. Our third speaker is Professor Dinesh Mohanji, School of Environmental Sciences, JNU, New Delhi. Dinesh Mohanji is a professor in School of Environmental Sciences at Jawaharlal Nehru University, New Delhi, India. He is an elected fellow of Royal Society of Chemistry, London, and National Academy of Agricultural Sciences, India. He is also an adjunct professor at Chemistry Department, Mississippi State University, USA. He was a visiting professor at Czech University of Life Sciences, Prague, Czech Republic, and adjunct professor at University of Southern Queensland, Australia. He has completed his master's and PhD from Indian Institute of Technology, Roorkee. Professor Mohan also worked as a postdoctoral associate at Penn State University and Mississippi State University. He published more than 170 research papers with a total citation of more than 45,000 and edge factor, which was 82 in the high impact factor journals. He has also authored 3 books. In addition, 2 patents have been granted to him. He has received global recognition as the Declarative High Cited Researcher in 2014, 2015, 2016, and 2017 till 2022. Professor Mohan has been the recipient of many academic professional recognitions, including 207 Scopus Young Scientist Award, Hiyoshi Environmental Award 2009 given by Hiyoshi Corporation, Japan, USQ 2017 Research Giant by University of Southern Queensland, Australia, Clarivate Analytics India Research Excellence Citation Award in 2019, and he has been named as an outstanding scientist by CSIR India. He's involved in various research activities, including water pollution, contaminant remediation using sustainable technologies, biomass conversion into bio-oil and biochar, climate change mitigation coupled with soil fertility management, contaminated soil remediation, and carbon sequestration in soils. For the first time, he has established a 3D concept of assessing absorbent potential of biochars, which is now widely recognized. Recently, he has developed a sustainable technology to solve the stubble burning problem in India. Our fourth panelist today is Dr. C. Arun. Dr. C. Arun has completed his PhD on garbage enzymes, where he studied production of garbage enzyme from pre-consumer organic waste and its influence on dairy waste activated sludge to enhance biohydrogen production. He is working as assistant professor at the Department of Biotechnology, Periyar Maniammai Institute of Science and Technology, Thanjavur. He has a teaching, research, and industrial experience. He has published many research articles on production of garbage enzyme and its application. I welcome all our panelists today on this amazing panel discussion which we are going to have. The discussion during this session will center around the role of bioenzymes in agriculture and how they can help in sustainable agriculture and food production. We hope to gain valuable insights into the potential of bioenzymes and their impact on agriculture, as well as explore potential solutions to the challenges faced in the field. So let us start the discussion with Sri Patanjali Jhaji. Welcome, sir.
Thank you so much. Distinguished scientists, eminent speakers on the dais, equally distinguished guests, extremely passionate about the work that you do. I would try to definitely limit myself to 20 minutes. A very bad speaker at one, but do listen to the bhav, not the language. It can come in very handy for saving us from mass distinction. For the last 20 years, we started this spiritual journey. My parents, my mother is no more, father is 95, they lived there on a desolate place in Madhya Pradesh next to Narmada Ji. And we made mistakes, and now we have established. I was very determined not to till the soil, very determined to use the trees in a different way, because this country has what we have in plenty is a sunlight. So I wanted to harness that. So the best way to harness the sun energy, I don't need to be a scientist, is through let it fall on leaves. Not one leaf, set of leaves. So this was the journey, and we would just go through some of the succulent and to the point presentation. This Goethe is, I'm a big fan of this philosopher. Everybody knows in the world that we need more trees, more grass, more covering. We are not doing enough. That's why the degree, now IPCC report says that 1.5 degree is imminent, not even 20 years away. Within 5 years, once it will cross. What are we doing about this? And there are many things which have become irreversible, including the rise in the sea levels, rise in temperature to a large extent, and greenhouse gas emissions are at its maximum. So, what is the solution? I'm a great admirer of this Indian scientist, settled in the US, Dr. Ratan Lal. He's a recipient of World Food Prize, which is equivalent of Nobel Prize in soil and agriculture. He says that merely 2% increase in the organic carbon content of the planet's soil can reverse all greenhouse gas emissions, all. It's incredible. So let's get down to it. How do we achieve this? Soil is the second largest depositor of carbon after the oceans. And currently the agricultural practices are contributing 30% to 40%. Estimates vary of greenhouse gas emissions. Nobody really, comes and claims that for obvious reasons. It's not the fault of the farmers. We have been, taught. They have been taught that this is the scientific way of growing. Gari jutai karo, matti badlo, dhoop mein sukhao. Everything which is just against the right method of growing. People say that the green revolution was such a humongous thing. That was the need of the hour. I don't agree with that. One of the gurus who I admired, Bhaskar Save Sahab, he was a recipient of many prizes abroad. But since he was very critical of the #1 family in the country and Mr. Swaminathan, so he was never given his due position. I was very fond of him, and so was he. So on 2 occasions that I met him, he made the picture clear. So I had just 2 gurus, one Fukuoka, one Save Sahab. I pride myself being the purist. What emerged? The simple innovation which we figured out over a period of time, and I have reverence to Mother Nature as God. So therefore, it is this. Grow 2 trees together. Independently, they are weak. They are the same picture from 2 different angles. One is moringa, one is bay leaf. Surround it with vetiver. Everything surround with vetiver, because it does enormous amount of carbon sequestration. I have very eminent scientists. I'm sure he would be aware of the capabilities of vetiver. It multiplies minimum 10 times every year. What I was mentioning in the morning, of 1.75 crore vetiver that we gifted to Eco Task Force of the Indian Army at 8 different locations, including Marathwada, including Gangaji, including Himalaya, including Rajasthan, has multiplied already in excess of 100 crores. The current Lieutenant General heading the Eco Task Force is also a friend. He has promised that in his tenure, he'll take it to 10,000 crore. One single act of donation of 1.75 crore by a group of 10 middle-class people would create more carbon sequestration than given by any government, given by any corporates. That is the power of Mother Nature. That is our great Indian Army. Coming back to the farming and growing. So yeah, this vetiver, why vetiver? You surround this. This is King of Thailand is the main patron of vetiver now. World Bank supports it. Every country in the world, including China, has adopted it except the country of its origin. It's a matter of great shame. Vetiver originated in India. So this is Chiang Rai Institute, Agricultural University. They surrounded two imli plants of the same age. You see the results. The one surrounded by vetiver has grown 5 times faster because it holds the topsoil, it holds the moisture, and it brings micronutrient from the lower depths to the root zone. So then again, 2 trees together, surrounded with vetiver. Inside the soil, off late, past 3 years, I understood black turmeric. Phenomenal thing to fight cancer. They say that 10% Indians are going to get cancer in their lifetime. 14 crores. One person gets cancer, everybody in the family is involved against the fight. And this is, I'm not saying, WHO says this. 56 crore would be struggling against cancer in a country of 140 crore. Is that something which is acceptable? No. Are we going to, where is the money? We will have some testimonials later on. If a black turmeric is kept inside the soil for 14 months, it has a curcumin content of 25%. It locates the cancerous cells and kills. There is nothing on earth which is better anti-tumor, anti-cyst than this. So we grow this. Perhaps we are the largest grower. Perhaps we are the largest, vetiver grower. And we take no money absolutely from anyone who is suffering in life. Some 300 odd people who had thought that they would never be cured in their lifetime have been cured. No evidence because in this, we'll come to that. Yeah, this is the Indian School of Mines test report in Dhanbad. 25% of 2-year maturity, 15% of 1-year maturity. If it is kept in under 14%, I get a sense that there are 7 crore people who suffer from rheumatic arthritis in the world. A significant number of them live in India. Most of them can be cured. This is brilliant against lungs diseases, brilliant against improving your heart condition, Alzheimer's, dementia, whole host of things, mother of all herbs. This is brilliant, and when grown in these conditions for 14 months, it's incredible. Coming back to this, what we do, the farming is multi-layer farming. The same soil supports, at different heights, different trees and plants. 2 trees together, suddenly if done on scale, we are talking of 100% productivity increase. And expenditure, 0. Nothing. Grow. Learn to give to soil, learn to be reverential towards Mother Nature. She is the most intelligent teacher. No one is more intelligent than her. Follow her. And so when you surround it with the vetiver, it is doing enormous amount of carbon sequestration, and you prune it and put it as a mulch. They again improve the microbial conditions. Mulching is fantastic. I'll also show some of the real pictures of our farm, and if I have to describe it, 2 trees together. We also grow. I realized those who have experience of being in the rural areas, the serious most problem is that of the ईंधन, लकड़ी. अभी भी गैस तो आता है, पैसे नहीं है उतने, तो स्मोकलेस चूल्हा है. और आने वाले समय में और भी गवर्नमेंट के बहुत अच्छे स्कीम्स हैं. करंट गवर्नमेंट इज done phenomenally well. Our Honorable Prime Minister is completely for natural farming. I thought we played some little bit of a role in that by meeting Vice Chairman Niti Aayog that time, 4 or 5 years back. He had not heard of Fukuoka, but he had an open mind. He visited our farm with his wife and with his sister. He asked me, Patanjali, what is the, how remunerative it is. Most people ask. We have two farms, one in Bihar, one in Madhya Pradesh. Madhya Pradesh is completely now spiritual, because what is going to kill the world and us human being is the greed. Nothing else. So if I also fall to that temptation, I'll be... So when I see that 100 tons of black turmeric growing in our kind of soil condition, I don't think that how much money would I get out of that. Instead, I feel that how many people can it cure. That's the money for me, that's the wealth for me. But at the same time, in Bihar, because we have farmers, I'll be blind if I do not say that what is the remuneration. It's so much that it is out of your mind. That what in agriculture, we do 7 layers of farming. Again, coming back to the farm, we also have started putting the vine of shatavar, of black turmeric also, because climatic conditions have changed in Bihar, and also of pan, beetle leaves. I know beetle leaves, again, whatever we were offering in our puja has a humongous, medicinal value for lack of calcium, or whatever you call that. That beetle leaf is amazing for sugar. It is very good for digestion. It's very good for many more things. It's very good. When grown on a moringa tree, it is even better. So that we have in plenty, and we know what is the price of a, one leaf of a pan. That inside that soil, the black turmeric. Some journalist. I was in a mela, meeting people generally. My best friend has Parkinson's. So we are fighting together with that, without the allopathy medicine. And in that, so I meet everyone. He's my dearest friend, and very, very much together in, in this whatever mission we have. And some journalist came, and someone was selling black turmeric, and so I was standing there, the same stall. She generally asked, what is the price? And the Vaidya said high price. So I said, no, I can make it available for 400 rupee a kilo. She was aghast, absolutely. Many people are selling for 3,000, 6,000. Even 400 rupee a kilo, if people can get, I'm not guaranteeing whether you will get it or not. But considering if you can convey this message that in 800 rupee you can be rid of your cancer, imagine the number of demand that it will create. Then grow it for 14 months. Good progressive farmers would do that. Farmers are the most innocent people. He has been taken for a ride, and among the farmers there are so many who are completely in sync with the best practices. Somebody needs to show them how to do it. Anyone who asks me, are you conducting a training? I said, rubbish. Why? I said, if you have not done your farming, come to our farm for 2 hours. It's embarrassingly simple to grow trees together, plant a vetiver, grow them, give it gay abandon, cover every inch of soil. In the year 1, the temperature comes down by 3 degrees. Mother Earth is burning. Where is the time for learning the permaculture, the dash disha, ye disha, wo disha, rubbish? Go vertical. 7 layers of farm. The top layer is subabul, grows fast, goes to a level and height which does not stop the sunlight. Beneath that moringa, beneath that agastya, amazing tree which also fixes nitrogen. In Maharashtra they know about it. In South they know about it, agathi. Beneath on that you have vine. Inside the vetiver, tuar, again for nitrogen fixing. Inside the soil, the black turmeric or elephant foot, or again biodiversity and whole. Our entire farming is about trees, bees, birds, and soil. So coming back to this, the cancer, the temperature, the water, the temperature, I'll wind it up in 5 minutes. I can get up from my sleep and can talk about it, because I've just done this and my job and nothing else. So I can understand. So there are 5 things which is plaguing us. Is the temperature. Temperature comes down by 3 degrees in the year 1, because most basic science, the sunlight does not hit the hard surface, does not bounce back. It gets absorbed, not on one leaf, on several leaves. Second is the soil. We have among the worst soil on earth today. because of the wrong practices. This makes the best soil on earth. Third is the recharge of groundwater. With so much of vetiver and so many roots, within 2 to 3 years, here we did something on 200 acres in Maharashtra on a hill. Normally at the downhill, the villages, wells would go dry, which now has water. I mean, beyond February, there used to be no water. Yes, simple things. This humble grass we have underestimated and not learned. Because they multiply by a multiple of minimum 10. And this is a perennial grass. This belongs to India. So temperature, water, soil, employment, combine it with the compressed biogas. So this government is phenomenal. They have understood the energy independence. So the surplus biomass, which is nothing less than 40 tons from the year 1, 100 tons in 3 years. At 3 rupee a kilo or 2 rupee a kilo to the farmer will make far higher sum than anything else. You grow many tomato, many potato, many other things at the same time, the prices crash, you have to throw it. That's market forces. There is insatiable demand for the energy. No matter how much you grow, there will be someone who will pick it up. Government has come out with a scheme of 10,000 crore investment on Gobardhan CBG scheme. That's a beginning. So, if we have cooperatives who, after usage of the biomass, we give it to the, the CBG plant, but it is not only giving it to the CBG plant. You receive that residue after producing the cleanest energy. You receive that residue, which is an organic fertilizer. Perfect circular economy. There is no science, there is no circular economy more befitting to our country than this. Grow in abundance, least bit of a cost, grow the best on earth, combat cancer. We will just show 2-3 testimonials for cancer. Or even better, I have a colleague, I'll end with that. She was former Chief Commissioner of Income Tax. I wish a mic can be given to her. Or if Ratna, if you can come here. Her husband had so-called, he was the Principal Chief Commissioner GST, had 4th stage cancer. A year back, they said, he's fine. Yeah, let Ratna speak to her. So I think since she participated here, instead of writing in words, let her speak. And then I'll conclude. Yeah, Ratna, please. Since I had the privilege of her visit here, I said, instead of my showing, 300 people so far have been cured who thought that they would not be cured. Of them, 35 of them were suffering from cancer. Yeah, Ratna, please.
So, black turmeric, for us, it's nothing short of magic. It's a miracle itself. This is a whole new world, this green organic world, which was introduced to us by Patanjali sir himself. We are from Bengal, we are very skeptical, we question everything. And so far as medical treatment is concerned, before this, we've never tried anything other than allopathy. Nobody at home believes in this. But everything changed when one day, suddenly, out of the blue, my husband was detected with cancer, and the world around me shattered. And that's when I met Sir. It's been more than a year now, and from that time onward, my association with black turmeric in particular, but even other stuff, vetiver, giloy, and all have begun. Initially, it was a challenge even to, make my husband consume these stuff. Like I said, we never believed in all this, and when the doctors say that there's no medicine for this because his cancer has spread, but now, that he has himself experienced the magic, this is the first thing he looks for every morning. And thanks to the extreme generosity of Sir, I have been able to give him this every single day, 1 year back till now. And it's so, I know, but, you know, this is something very rare. We don't get it anywhere. And it's, I don't, and since Sir doesn't charge anything for it, it's absolutely free. In fact, I don't even have to ask for it. A colleague of mine will say that Sir has sent this for you. So I have been trying to find my own avenues of getting black turmeric, because it is really, you know, it's too much, keeping on asking him for this year after year. So only once I have seen this black turmeric in Nature's Basket. These small packets they were selling at ridiculously expensive prices. If you go by their price rate, it comes up to 60,000 rupees a kg. 60,000 rupees, and that also I placed an order for that, and it's been 2 months now. They have not been able to come back with that. They said it's very rare. And what Sir gives us is the kind of black turmeric which has been in the soil for 14 months, which I believe works best for all kinds of cancer. And one thing that I have seen is that, you know, with God's grace and Sir's help, till now, my husband is far, far better than he was a year back. And more than that, I have seen that in the initial stages, he would continuously catch infection, something or the other. It was either UTI, or it was a sore throat, or it was a lung thing. These things have completely taken care of that. He has not had any kind of infection over the last 6 months. And like I said, that for us, it's, you know, Amrit. What can I say about Sir? You know, rarely, if ever, one can get a person like him. He's an embodiment of spirituality, of humanitarianism. I run short of words, you know, to describe his qualities. There's nothing we can ever do for him, but he's always there in our prayers. And I hope that he achieves his dream of 100% green organic cover for India, a green world that heals and cures. Thank you so much, sir.
Thank you, Ratna, for kind words. I'll just sum it up for my farmer friends. Abroad, it sells at incredible prices, 5,000 rupees a kilo to anything. And so we need to convince our leaders that our Prime Minister, Honorable Prime Minister, is so keen for the doubling of farmers' income. It can quadruple. So no cost, these things. The whole world is full of cancer. The whole world is full of rheumatic arthritis. That's one. Vetiver water is very good for your body. And all these have great money. I don't grow vetiver for oil. And the best thing which the current government has done is about bringing the Mota Anaj center stage for the first time. It is incredible. All of them can be grown by broadcasting without tilling. And when grown this way, they are medicine par excellence. Thank you so much.
Thank you so much, sir. We are indeed blessed to have you amongst us. In this Kaliyug, people like you are very rare to find. So there's one small announcement to all our speakers. We are actually having a time crunch over here. So I request everyone to limit their time to just 5 minutes, please. And our next speaker is Dr. Susan Titus, ma'am. Ma'am.
A very good afternoon to all of you. Well, first of all, let me bring warm greetings from our laboratory, Naval Materials Research Laboratory, which comes under Defense Research and Development Organization, which is situated in Ambernath. And I take this opportunity also to thank the organizers for having invited me for this G20 Summit conference today. Thank you so much. We are all talking about, in this conference, about the environment, sustainability, and bioenzymes and all. So we are all concerned with the environment, right? I hope after a sumptuous lunch, you people are not very sleepy or anything. But let us if anything you want to ask me also, but I'll finish it within 15 to 20 minutes. That's okay? 5 minutes, okay. So I'll just skip all these things. Please, next, please. This I want, the primary duty of every human being. We should be thankful that God has given us a very beautiful environment, so it is our duty to protect it, to sustain it for the next generation. So this should be our motto, whoever is assembled here, and also to take the message. As Sir has just now given a very good message and a very good this thing for each one of us, so we should carry this message with us. Next, please. So next, again, the same thing it is. You can see how our earth looks. It's crying if it is not, if it is polluted and if it is not sustained properly. Next, please. So I'll be talking about marine pollution, in fact, because we are connected with the Navy. We are doing research for the defense, that is the Navy in particular. So I'm in Marine Biotechnology department. So here actually, well, we can skip this, but main thing I would like to tell you is that we may think that oil pollution will not harm us, but no, my dear friends, because they contain this hydrocarbon contaminants are recalcitrant in nature. That is, they're non-biodegradable. They are persistent toxic chemicals which are there in the environment, and these are highly carcinogenic and mutagenic. We may be thinking, even now babies of 6 months and 7 months, I know my friends, they are having cancers, and they have been detected, and you may wonder from where they have got this. This has been inherited from their parents, because this due to bioaccumulation and biomagnification, that is how through the food chain it comes, and it is causing great havoc to the public health. Next, please. Navy. All the naval commands are using this technology of ours, and also we are now told to commercialize this and even put it for the other ports and everything. Next, please. So next, please. Again, this is all. Everybody, just a general thing it is. Please, next. So this all we know. I have already, you people, since I've given only 5 minutes, so I should maintain the timings. So here, this actually remove oil spills. Still, if you see, bioremediation methods or the natural methods are not in very vogue. Why? What is the reason? Because they are very time consuming. Because even if you see the US EPA products where they are listing around 9, although several are there, even our TRI is also there, Tata Energy Research, they also have come out with the oil zapper technology, but that is not meant for the marine pollution. So there, what is the time consuming? It takes a lot of time. Even the EPA, which I just now mentioned, the 9 products which are listed, they take minimum 7 to 10 days. And that is a major drawback of this. Although it is purely natural and it is very good for the environment, safe for the environment, people are still resorting to the chemical dispersants where it is highly toxic, and it is also even now recently BARC has also come out with a technology like they have an absorber which can, a polymer which can absorb all the oil. But it's just like for reusing the oil it is okay, but it is a temporary solution, like you're collecting it from one place and you're not for the pollutant oil or the weathered oil, exactly. So, but for if the large oil spill is there, they may be good. These biosorbents I'm talking, not the biosorbents, the like sawdust and glass wool and all. But in case of physical, they still, the Navy is using skimmers. They have, for the oil spill response, they have the skimmers and all, which are, clumsy and also very laborious also. Next, please. So the challenge is to come out with a rapid as well as a safe technological solution, and to create a clean and sustainable environment. Next, please. So this bioremediation, I think so everybody is, they know about this terminology. That is, we use biological organisms and their products to remove the pollutants from the environment. That is, these, of course, we should thank the nature or God himself, because who has endowed us with a one-celled wonder microbe, which has got the wonderful capacity to degrade or to consume all these pollutants in a marvelous way. Next, please. So these are the main components. They are the microbes, I said. I think so it has been a little bit shifted. So anyway, Bioremediation, like these in our technology also, we have made use of these four things, biosurfactants. Why biosurfactants? Because you know that oil forms an impervious layer on the surface and it does not mix with the water, it is immiscible, and therefore we need certain surface active agents and biosurfactants which can emulsify this and bring it into the water layer for the degradation by the microbes which are already present in the water layer. And also efficient microbes and oxygen for oxidizing these hydrocarbons. That is the main thing, because they are just hydrocarbons. When oxygen enters, they can be easily metabolized by the other organisms in the water. And of course nutrients. Nutrients is a very important thing, because when you are having this carbon in large amounts, what happens is the nitrogen and phosphorus becomes limiting. Same with us also, in our body also. If we keep on taking carbohydrates only, we'll start developing deficiencies. So this, for utilizing this carbon, they need nitrogen and phosphorus for the making of enzymes and DNA and all this very fast. So that we have to supplement them also along with them. Next please. So I'll be just now showing you the NMRL, that is the Naval Materials Research Laboratory, the technology which my department and me as a PD we have developed. So it is one of which I told you, it is very innovative as well as it's totally indigenous. Everything right from the scratch to the bottom, we have done everything in our laboratory. We have not taken help of anybody. Next please. So our approach is nothing but very simple. We have developed two products, Biomix and Nutrimix. And this combination of this Biomix and Nutrimix in the presence of oxygen will convert this pollutant oil into fatty acids, fatty aldehyde, that is oxidation of these hydrocarbons, and the increase in number of the cells of bacteria. They also multiply. Ultimately, they will be going into the Krebs cycle and beta oxidation pathway and all, and they will be converted into carbon dioxide and water. Next, please. So we have in our laboratory itself, we have microbiologists, molecular biologists. We have a very well-equipped laboratory in Ambernath, and where we have brought from the seawater itself. We have an access to the naval dockyard, Mumbai, and we have gone there. My team has gone there. We have collected the oil-contaminated waters, and we have several bacteria, around 30 to 40 bacteria, marine bacteria, we have isolated, and we have done several screening assays, and we have come out with 3 potent bacteria which solely survive on oil. They do not grow on any other carbon sources except on oil. As long as the oil is there, they survive, and otherwise they die off. Next please. So next, yeah, these are some of the characteristics of these bacteria. We have done several of studies. The whole project report is there, but I'm just showing you which is just for this particular conference. We have the bacterial adherence to the droplets. We can see wonderfully, you can see the acridine orange, the fluorescent microscope pictures, where you can see small bacteria adhering to the oil, and also emulsification activities with each, either long chain, short chain, and even middle chain hydrocarbons, even aromatic hydrocarbons. You can see they emulsify these hydrocarbons. They have a surface active property. And also we have done the molecular biology studies. This shows that these bacteria are endowed with genes which can produce enzymes to degrade long chain and short chain, as well as aromatic hydrocarbon chain hydrocarbons. So that is the beauty of these organisms. They have multi-degrading hydrocarbon genes in them. And this non-hemolytic assay is that they are very safe. They are non-pathogenic. They do not lyse the red blood cells. Next, please. So this is one of the products, Biomix, as I said. They are the consortium or a mix of a blend of 3 different oil-degrading bacteria, which we have isolated in our laboratory, and which is easily available in the native waters of the Mumbai. And we have now patented these organisms also. And these are some of the properties. Next, please. The next is Nutrimix, as I said. Nutrimix is also another product. These 2 products are the ones which, when combined with 15:1 ratio, when we spray uniformly over the oil, you can get the degradation as well as the disappearance of the clearance of the oil. Next, please. So the beauty of this Nutrimix is also there. Many people have come out with this nutrient compounds also, but in that, they have this very toxic one, butoxyethanol. Even Inipol, if anybody is interested in this, if you see Inipol, the Exxon Valdez oil spill was there. There they had made use of the Inipol, and still actually the Coast Guard is also recommending, the nutrients only. But they contain butoxyethanol, and now this butoxyethanol is banned actually. They are not to be used. It's just not our finding. In fact, we have to go through several stages when our technology technology is accepted by the Navy. So we have done several validation, and this is one of them in the boom where 30 liter of seawater was taken, and we have just put this around say 1 to 2 liters of the, it's fully covered with the pollutant oil, that black thing which you can see. There's no thing. You have a this thing, what you call laser print thing, pointer, sorry. Okay, no problem. You can see the first one where control is written. You can see it is completely filled. It was like this. And then later on, we are providing aeration through the stirrer. And we have poured our biomix and nutrimix on them. And just after 12 hours, you can see in the test where our bacteria and the nutrimix or the biomix have been put. You can see that they are degraded, and you can see that oil is removed. Whereas in the control, if you put your, how to tell, but of course GC we have performed, and also that is also there. But here, like we put a gloved hand, you can see that oil is sticking onto the glove. Whereas in the case of where we have put our nutrimix and biomix, you cannot see that. So this itself is a proof that all our this thing has been degraded. Next, please. So here also we have, because when Navy had come, they also said, you please put a filter paper and let us see. So this is the results of that. You can see in the control, you can see that the filter paper has the oil has adhered onto the filter paper very badly. Whereas in the case where we have treated, it is absolutely clean. Next, please. So this is also the GCs we have performed along with the ICT. Also we have we had given them a CARS project to the ICT team also, and they have performed all our GCs. In fact, we also have a GC everything, but they were also told we have done on payment basis. We have done with them this GC of this thing, and they have because they have proved and they have done several of our GCs of our residual oil, and they have said that 94 to 96% is degraded within 24 hours. Next, please. So this is some of the field trials we had to do, and field trials also we did some 2 to 3 time field trials. Means we had to go to the naval dockyard, to the field, and we had to like they had collected all the pollutant oil. The pollution response team of the Navy, they had collected the Western Coast, Western Naval Command here in Mumbai, and they had collected these pollutant oil, and they had put it into the boom that which is they had contained that. And then we had, you can see the spray. This is just one step spray. Normally what happens, there's a sequential spray also. Like many of the technologies whichever are available, bioremediation technologies, they spray in sequential manner. But here in this case, we mix in the ratio 15:1. And this is just a one step spray and very simple. And we can see that within 24 hours, there is here emulsification, and the first thing which happens is that because of the biosurfactant and the degrading nature of the bacteria, they start degrading and they start dispersing the emulsifying the oil, and within 24 hours, next please, you can see that there is complete removal of this oil. I have a video also, I think so, I'll show you that. Next please. So here also we have taken out water samples from the field, and here also we could see that most of the hydrocarbon peaks had been degraded. Next please. So this application method of ours is very safe, and the Navy has really, they have appreciated our technologies, and for the past 2 years now they have using these technologies. I mean, Mumbai is using now all the naval commands, that is the Southern Naval Command, Andaman Nicobar, Karwar Waters, and our Eastern Naval Command, Vizag, Cochin, all these people now they are using our technology. We have transferred the technology also, and it's very, very cheap. No costly equipments are required. You may be thinking the bacteria just requires the pollutant oil, 1% pollutant, and it requires the what you call the nutrimix in 0.1%. That's all, and it's very cheap. And I mean, you can there are various ways to preserve this culture, and that I'm not going to. Next, please. So can you please switch on that video? This actually, I had the full video, but this I'm just showing the field trial which we had done on the naval dockyard. So here, this is in the South Break waters in the naval dockyard, and you can see me standing there also, and the Navy person also there. And this is near the INS Vikrant. They had given us the site where a lot of this thing, and see here my team and the pollution response team and their people from the naval dockyard, they had helped us, and we are spraying. This is just, we are spraying our mixture that as I said, 15:1 over this oil. And soon after this, our mixture, our bacteria and our nutrimix, they adhere onto this this oil. That is, they don't get solubilized, and they immediately disperse and disintegrate this oil. And you can see that they have dispersed and emulsified very nicely, and you can see when we are spraying our reagents itself, they are breaking into droplets and they emulsifying and dispersing. Otherwise, oil being, what happens, they come and get collected at one place. That is not happening. They disperse, and within 24 hours, in fact, we started at 10 o'clock, and the next day when we started at 1 o'clock, next day at around within 24 hours. We have just kept 24 hours. But yes, this was a real achievement, and the Navy, the Chief Admiral, they have also appreciated our technology, and our technology has been listed as one of the eco-friendly technologies also for them, for green environment. And still they are wanting us to now for all the ports which are available in India, as well as in for the Coast Guard also, for the initial oil spill response. Next, please. So this is the advantages. I need not tell you. The main advantages here is that it is very cheap, very safe, very useful for the environment. It sustains the environment, and also it is very easy to produce. And best thing is that which is not there in the whole world. In fact, I can say it is not available. It removes the oil within 24 hours. You will not have any technology, eco-friendly technology, which can remove within 24 hours. Minimum is 7 to 10 days. Next, please. So this is our status. We have already, technology has been inducted by the Navy, as I said. This is our policy now, this thing, and 2 patents we have filed on these products. And technology, we have already transferred this first to the defense category, that is A. That is category A is only for the defense. That is for the Navy, but still, because of its demand and because of its usefulness, we have now also transferring to the commercializing this. And there is another partner who has come forward, who is who is into the Coast Guard and also all the ports of the Mumbai, as well as outside Mumbai. And this product has been realized, and it already has been supplied. We have supplied ourselves for 1 year for the Navy to really adopt this product and to get used to this product. And then after that, we are now, our technology partners are helping us. So next, I think so I have done with. This is just a spin-off of that technology. Now, since we are, a lot of people are here presenting on biosurfactants also. So this bacteria, they are also having a lot of surfactants in them. Apart from that, when we say the word bacteria, you know how people are, if they are not from the biology background, they start getting scared. It may be having some pathogenic effect, or it may be causing some disease. So we have also taken a herbal-based product, and we have combined these two, and we have done, we have come out with a bio oil cleaning solution, which is now we have already done we have already demonstrated to the Navy in our laboratory, as well as we have done on a small scale. We have done on INS Mumbai, which is one of the ships where my team had gone, and they are very satisfied. And now we will be also transferring this technology. Next, please. So this is also just a video. I think so. After this, I'm done. Yeah, please. Can you just show us the video? Yeah, that is the vision. I said this is also another message. Our vision should be. You can show this. It's not very big. So here actually it is showing like how our product, when on the oil, we are just applying it on the surfaces where it is oil. We have created that oily surface, and we just apply this lightly. We rub it after say 15 to 20 minutes, and we wash it off. In case if it is still there, you can have a second application. But of course, we have come out with all the how much we need to apply and all that. So yeah, we can stop after this, and then because it need not go to the other one. Yeah. So this message I would like to tell was that please protect and present our future generations from various environmental hazards. So please, all the students who are here and all the scientists who are assembled here, please, that is my humble request, that we need to work for our environment and also for our generation. We need to love our environment, and we should promote sustainable practices and shift towards friendly environment ways. Thank you all. Next, thank you. Can you just show the thank you slide? Yeah, so this is actually how we have gone, and several, even you can see our chief, this admiral there, and our Dr. Satish Reddy, all of them, they had come and they have personally, seen, and after that only we have been able to induct this technology. Thank you very much. God bless you all.
Thank you so much, ma'am, for the insightful session today. Our next speaker, Professor Dinesh Mohanji, I request you to please come up on the stage, sir.
So good afternoon. I won't take much time, maybe 20 minutes will be more than enough. So I will try to give you a basic idea about this concept, what we call biochar, and that concept can give you the answer regarding the sustainability of the biochar for many applications, including burning of the biomass, then food security, soil fertility, water purification, and also the climate change mitigation. So I will not be talking about the goals and everything. Let me give you some idea about the solid waste in terms of the agriculture residue we are generating every year. So see, we are getting about 600 million metric ton, huge amount of agriculture residue every year. And most of the agriculture residue we are burning without any use. The highest producer is Uttar Pradesh, followed by Punjab and Maharashtra. So we have a lot of agriculture waste which we can use for some important or useful purpose. And this waste is being burned everywhere. It is not like we are burning especially in and around Delhi, but if you go from south to west to east to north, what they are doing is they are just getting the food. After that, to clean the agricultural land, they are burning. So when they burn, I think we are getting a lot of contaminants in the air. Everybody knew about that. But just to give you an idea, when you burn about 1 ton of stubble, you are putting about 2 kilograms of SO2, 3 kilograms of particulate matter, both PM2.5, PM10, 60 kilograms of CO and C here, 1,460 kilograms of CO2. So huge amount of carbon dioxide we are putting in the environment. So there are many factors why farmers are burning this in the agriculture field. So I summarize some cascading factors, including social, agriculture, political, technological. So one of the important factor is, like from one crop to the next crop, they are having only maybe 1 week or 10 days. So the best solution is you burn it, and they also have some impression that when you burn it, you will be getting some nutrient in the soil. That is not true. When you burn, you are also losing organic carbon, and lot of properties you are losing from the soil. So what we are trying to do is, we are trying to find a solution for this agriculture residue to convert into a useful product. So this is your waste. Any waste where you have the organic content, you just take it in place of burning. Either you do the pyrolysis, you convert it into the liquid we call bio oil, or you do the gasification. Many industries are doing, and they are making syn gases and converting into the electricity. But finally, the process of doing the pyrolysis and gasification is difficult. So what we are doing is, we are trying to make it into a useful product called biochar. This biochar is a porous material, high surface area. So you can use this biochar in the removal of contaminants from water. This also improves the soil health when you add that in the soil. And finally, when you put in the soil, it is going to sequester the carbon. Now, depending on the process, you can also increase the biochar yield. If you want to go with the less biochar, you can go with the fast pyrolysis. Or if you want to go for the high yield, you can do the slow pyrolysis. And of course, for gasification, you need to do at very high temperature. So this is a process where you need to put a lot of energy. Now, what is biochar? So biochar is a fine-grained charcoal. I think everybody is familiar with the charcoal. This is having high organic carbon, and this is largely resistant to the decomposition. So the half-life of this biochar is 1000 or 2000 years. That is well established. Now, if you see the major property of this biochar, this is a carbon negative technology. The other processes, if you are taking the biological processes or other thermal processes, either those processes are carbon neutral or carbon positive. But this is the only technology which is a carbon negative technology. Means you are not only stopping the CO2 going into the atmosphere, but you are also drawing some CO2 and keeping in the soil. So this is the major advantage of this biochar. This is having high surface area, very high water holding capacity. This is stable up to 1000 years. Now, this also increases the microbial activity. So those who are interested in the microbes, I think we are listening so many things from the morning. They can also try using this biochar in the soil and see how the microbial activities are changing. And also, they are going to retain the nutrients. So I will demonstrate you one example where we have seen how the soil properties or soil health increases. So why it is carbon negative? Like everybody is familiar how we have this CO2 taken by the plant, then you have the plant death. So whatever CO2 or nitrous oxide is absorbed by the plant, it is going back into the environment, so it is a carbon neutral cycle. When we are using the fossil fuel, we are putting some additional, so that is carbon positive cycle. So what we are doing here is, we are taking some of the biomass, converting into biochar and putting in the soil, so that it can stay here for 1000 or 2000 years. So we are converting this carbon neutral cycle into carbon negative cycle. In fact, in 2018, IPCC recommended 6 technologies to be used for the carbon management or CO2 sequestration, including afforestation, deforestation, then soil carbon sequestration, and many others. But they kept this biochar on the top because this is a low-cost, sustainable option. This is well proven. You don't need to do many experiments to use this biochar in the soil. Recently, in 2021, again IPCC recommended this biochar. This is mentioned maybe 59 times in this document if you see, and you will get many additional properties in addition to what I am telling you. So what is biochar? Many times we have the confusion that whenever you have this biomass, you convert into char, you will be getting biochar. But you have to be careful because you need to look into the atomic ratios of H by C and O by C. So usually biochar material that obtain H by C ratio higher than 0.7, we never consider that as a biochar. In addition to that, based on the O by C ratio, you can also determine the stability of the biochar in the soil. Like here, if the O by C ratio is less than 0.2, it is very stable and the half-life is more than 1000 years. If O by C ratio is between 0.2 to 0.6, moderately stable, it can stay up to 100 to 1000 years. But if it is less than 0.7, maybe you have less time. It is going to stay for a shorter period. So based on that, one can design the biochar we call engineered biochar or designer biochar. Whatever is needed, you can play with the temperature and the biomass quality, and accordingly you can manipulate the surface properties. So here I started this work. Basically I consider this as my baby, right? I started this work long time back in 2005 when we were making the bio oil. We converted biomass into bio oil and we got this char in addition to the bio oil. At that time I thought that we should try to use this in the soil or in the water. So we started with some experiments in water. So we published our first paper. This is the first paper where we used this biochar for the water remediation. At that time, no paper was published on biochar in either area. But after our publication in 2005, you see now we have the exponential rise, and now we are getting about many hundred papers per week or per month. So it is gaining attention of the researchers as well as the policymakers. So this is the first reactor we made, where you can just process the biomass at temperature between 400 and 500 degrees centigrade. The capacity of this reactor was 1 kilogram per hour, means in 1 hour you can generate about 1 kilogram of the biochar. Later on, we upgraded this reactor and we converted now, or we upgraded to 7 kilogram per hour, and we also kept one option where you can also get the bio oil in addition to the biochar. So depending on the temperature and the residence time, you can play with the quality of the product or byproduct. Now recently, keeping in mind the farmer problems, because they used to burn, especially in Punjab, Haryana and UP, because I'm in JNU, living in Delhi. So we have very, I think, difficult time during the winter when they start burning the residue. But it is not because of them. There are many other reasons, so I'm not going into that debate. But finally, they need some solution. So based on that, we made some reactor using the locally available material. So we developed using brick and also the kacha reactor, where we use clay soil. So the concept was like, once they are done with the harvesting, they can make the bigger reactor using clay in the agriculture field. And once they are done with the biochar, they dismantle it, because we have demonstrated when you have the clay soil, this biochar is more effective. So they can mix this clay and biochar in the soil and they can put the next crop. So we patented this item, this reactor, and we are also I think giving training to many farmers in Punjab and Haryana. These are the factors which are responsible for the overall biochar quality, means what type of biomass you are using, how you are keeping the temperature, heating rate, mass transfer rate, residence time, then what are the pressure, what are the gases inside the reactor. If you want to do some pre-treatment, let's say you want to develop some material for the water remediation, you do some pre-treatment, enhance the cation exchange capacity, and then you reuse it in the water remediation. So many things you can do to modify or to change the surface properties. At that time, we published a review article. If somebody is interested to get more information about that, this is about 60-page long article. You will get the designing part, mechanism, chemistry, and also the properties published in Energy and Fuels. So if somebody is interested, I will be happy to send that. Now then we applied this biochar in 3 different compartments. One is for the water remediation. So I will not focus on that, but I will try to give you one example how we use this in the soil. Let's say in the water, we developed even the cartridges, we developed some simple filtration unit, especially in those areas where we have high arsenic and fluoride. You don't need any electricity. So we made these cartridges using the biochar and by doing some surface modification, and you don't need any electricity or external energy. So under gravity, you will be able to do that. So different type of biochar we use and we applied for decontamination of water. Now let me give you the example how we use that in the soil. So what we did is we did many experiments, still experiments are going on. This is just to demonstrate you that in one small garden available in our school, we use biochar at the rate of 8, 24 and 48 tons per hectare as per the agriculture practices. Then we also use the biomass exactly with the same particle size and same dose. And then we also took one control. And then we incubated this for several days. This is in the lab and this is in the field. let me tell you, if your biochar is having about 90% carbon, though it is difficult to get 90%. Let's say you have biochar having 90% carbon. So if you want to convert how much CO2 you sequester or how much CO2 you put in the soil, you convert it, you will be getting about 33 kilogram of CO2. So you can imagine that if you are putting 10 kilogram of biochar in the soil, you are not only improving the health of the soil, you are also getting this CO2 out of the atmosphere. And because of this, you may be getting the carbon credit. So this is the additional advantage which you will not be getting in other processes, biological processes. So these are the simple experiments we performed to check the CO2 emission with biochar, without biochar, in the lab and also outside in the garden. And this is the simple experiment. We designed this reactor, a small reactor, and we put this biochar and soil together, and we measured the CO2. So this is automatic machine. So when you have this CO2 chamber, you connect with the pump to make the circulation inside the chamber. Then you connect it with the CO2 detector, and directly you will be getting in computer the readings what you are getting from the analyzer. This is our setup in the JNU. So let me tell you, this is very interesting. When we use biomass, we have the CO2 emission, right? The red tower is giving you the numbers when biomass is being used in the soil. And when you converted this biomass into biochar, you will be getting around 92% reduction. So this is an additional advantage, that less emission will be coming out of the soil or the agricultural land where you are putting your crop. In this table, I just try to demonstrate that how the soil properties are changing. Let me give you a simple example. When you have organic carbon in control, this is about 0.016, very small amount. But when you change it to, let's say, 3% of biochar, you will be able to increase it to 21.8%. This is for the organic carbon. Water holding capacity, only 11.2 without any biochar, without any biomass. And it is going to increase to 30, huge increase. So what this indicates, when you are using this biochar in the soil, you need less water. This is also having high cation exchange capacity. That means you need less fertilizer, because biochar is going to hold the nutrients for a longer time, and it is going to release slowly. So it will be more beneficial versus when you are using the fertilizer. These are some of the pictures. If you see, this is with control, this is with different doses. You see a huge difference. So I'm not going into that detail. This is number of leaves, plants. We have done all sort of analysis. These are some of the pictures where you will find, like, this is control and this is with the biochar. These are the eggplant. So excellent difference you will find. Even if you see these tomatoes, this is control, this is with biochar. We are not adding any other fertilizer or compost, only we are adding the biochar. Now see the fruit quality. When you take this eggplant from the after harvest, you see most of the time we are getting fancy and US grade number 1. But when there is no biochar in the soil, where we have not added any biochar, we are only getting US number 2 and Culsis. So it is also beneficial that the fruit quality, what you are getting, are very good. So you will get better price. Also, we are also in the process to characterize the nutrients available in the fruit. So I think very soon we are going to publish that result. This whole work we have published in RSC Advances. If somebody is interested, I think he may try to look into the paper. We have also done the carbon sequestration, how much is sequestered in the leaves, stem, root. So we did a lot of calculation you will find in this paper. Now, recently I was invited by Nitin Gadkari ji, and he was explaining to me that in Maharashtra especially in the area he was talking, Nagpur and Vidarbha, the organic carbon is going down and down. And he was telling in some places we are not getting even 0.2%. So if we can double even to 0.4 or 0.6%, I think farmers will stop doing the suicide. So he is very keen, he is very interested to have this project going on in Maharashtra. And he also suggested that if we can do the conversion of the bamboo, because he is now interested to have this bamboo and conversion into biochar, and if we can do that. So we are also doing some preliminary studies in our lab, and very soon we will be reporting to him. Later on, he also suggested to give training to the farmers. So based on that, I am also doing some sort of a training program. He is also the MP from Maharashtra, and Mr. Pasha Patel. He is also bringing many farmers. I am demonstrating in my school, in my laboratory, how the things maybe change when you are adding and how you can manipulate the data. This is one of the studies where we put the biochar in the contaminated soil. So it is very interesting. Even if your biochar, your soil is contaminated, mainly if you have heavy metals. So what biochar did is, biochar converted the bioavailable fraction into non-bioavailable fraction, and it is not going to release in the soil. So you have the immobilization of the metals. So this way you can also do the soil decontamination using this biochar. I'm not going into the detail, just showing you the application. So overall, this biochar is going to increase the nutrient availability, microbial activity, soil organic matter, water retention, crop yields. Then it is going to decrease the fertilizer needs, greenhouse gas emission, nutrient leaching, and erosion. In addition to that, let me tell you that if you are making compost, so we have published and we have demonstrated, if you use this 5 to 10% biochar during the composting process, then you will be getting less smell, and the nitrogen leaching problem, what you face from the compost, will not be happening. So try to use that, try to add this biochar during the composting process. I think you will get better product. In addition to that, in many countries, Germany, US, they start giving this biochar, 1% biochar to the cows or animals, right? And what they have reported is, when you do that, you have the less methane emission. So this is another advantage. This is also given as a food to the animals. Now you can also change this biochar into the biochar barricades, and then that can be used for the energy generation, another application. Now this is also being used to preserve the food. If you keep the fruits in the biochar, the half-life of the fruits will be increasing. Another application. So many applications now people started using in making the board. When you are making the doors, they are putting some biochar, and they demonstrated that the strength of the board increases. They are also putting in the cement. Once you put in the cement, half-life is 2000 years, so it will be there. You are putting CO2 and making it stable. So many applications. Market is also, I think in India, though we are having very less quantity of biochar, but in other countries it is expanding, right? There are many players in the market. They already started selling the biochar. In India, 2 or 3, I think if I remember correctly, 2 or 3 companies, they are producing the biochar and they are selling that in India. I remember one in Rajasthan. And these are international players. So I think the future of the biochar is excellent, right? Last one point, let me tell you, if we use the biochar, it is estimated that renewable energy fuels in 2100, whatever you are using, and biochar sequestration, if you take into account, if you establish that, that is about 5.5 petagram carbon per year. So you will have the equivalency. So whatever you are getting out of the fossil fuel, you can do the neutralization by putting the biochar in the soil. I think with this, I'm going to stop here. If you have any question, I will be happy to have the discussion after the lecture, right? Or maybe if you have. You can just Google it. In Ankur or something in Rajasthan. I don't remember the name. I am not producer. I am not producer. I am professor. I am a master. I am a teacher. But I am giving whatever I can give. I am ready to give the technology. I am ready to give the training to the farmers. And I am getting support from the government. And I hope in the near future, this will be the item, I think, which farmers have to adopt, right? And when you have this kacha reactor, you don't need external energy. You have to start burning it. And then we have the technology how to control it. You have to stop that burning immediately after a few hours. Then you have to keep for 6 to 8 hours, and then your biochar is, I think, ready to use. Sure. I am giving, Mr. Pasha Patel, he is MP from Maharashtra. Yeah, if you remember. Yeah. So I have given training to many farmers coming from Rajasthan, even Maharashtra. So he brought many times in groups. But I will be happy to give the training even in the villages, right? Yeah. I think online is not going to solve any purpose. You have to, I think, demonstrate how to control it. That is the main thing. So briefly, I told you what I can in 20 minutes. Thank you so much.
You can take it later.
Yeah, can you? We'll share sir's number with you.
My contact number is, you can share.
We'll share later. Yes, we'll take questions, sir, later. We have one more speaker to go.
Yes, tell me.
I'm getting 30 minutes or 20 minutes or 40 minutes, and it should be question asked should be coming. Because 40 minutes of talking about something, and people have a question inside. It cannot be on email.
Can we take questions?
I will be happy anytime, after, now, or whenever.
We'll take questions from you, and then we'll just request sir later on to interact with you, if it's okay with you, because we've got one more speaker to go. We respect everybody's time, and we respect your time as well and your opinion as well. So we'll proceed with the next session. Thank you so much, sir, for all the knowledge you shared right now with us. Our next speaker is Dr. C. Arun. Sir, please.
Thank you all for the gathering, and I am thanking the organizing committee for giving the opportunity for me to explain about how I have started the garbage enzyme production and how I have used it for sludge degradation. So my topic is based on garbage enzyme production and its characterization, because when I started in 2012, there is no characterization how to characterize the garbage enzyme and what are the things it is inside. We have not, technically we have not have any of the data or papers related to it. So when we started first formulating it according to the procedure what Thailand scientists have told. Then we have went for characterizing it, and then we have used it in an environmental application and sludge degradation. Next slide, ma'am. So generally we have enzymes means it is a biocatalyst which is essential in different biochemical reactions and different metabolic pathways, and it have two major things. It is intracellular and extracellular. In enzymes of bioenzymes, we thought it is most of them are extracellulars like hydrolytic enzymes, protease, lipases and amylases. So we have focus on extracellular enzyme. Next, which have play an important role in remediation and environmental remediation and biodegradation in environmental applications. So generally enzymes can be used in different applications like bioleaching, biosorptions, and bioengineering, bioaugmentation process. So these are the general how the enzymes can be used and how it can be help in reducing the load or the pollution in the environment. Next slide, ma'am. So generally the waste generation is around increasing every day because of the industrial sector increases, and as a result of population increase, the industrial sector also increases, and the commercialization of the product starts increasing. Even we are having instant processing foods. So all these things needs a water for doing the process, and a lot of wastewater is coming out as a effluent from the industries. So we focus on industrial effluent and how they are treating it. Next slide, ma'am. So this is the general wastewater treatment every industries may have. They will have a primary treatment, and then they will go for a secondary treatment where the biological process will happen. Then it will go for a tertiary treatment for purifications like RO, carbon-based sequestrations. Everything will be done in tertiary process. But when we are going for a secondary process, that is aerobic or anaerobic degradation process, there will be a huge sludge will comes out of the sedimentation tank. That sludge should be managed before it should be disposed into the earth, because it is having some pathogenic microbes, and that will affect the soil, and it also affects the atmosphere. So to control that, we have started trying what are the things we can do. Next slide, ma'am. So for stabilizing the sludge, the best process is anaerobic fermentation or anaerobic digestion. In this, 4 major steps will happen. One is hydrolysis, second one is acidogenesis, and third one is acidogenesis, and fourth one is methanogenesis. So we have found this hydrolysis process is a major rate limiting step. There the long chain proteins will be converted into shorter amino acids, and long chain lipids will be converted into short chain fatty acids. Then these fatty acids and amino acids will be utilized by the microbes. Then it starts growing, and it is converting into acetic acid, and this acetic acid will be consumed by the methanogens, and then it will be converted into methane. So for that they need some environmental parameters. So we have found these are the steps which is happening in anaerobic digestion, and the major rate limiting step is hydrolysis. So we want an enzyme which should be reduce the process of hydrolysis. That is, that protein should be converted into amino acids. Then if it is in a ready-made stage, then it will be directly the microorganism will start consuming. The lag phase will be reduced. So to do that process, we have done we have taken some of the enzymes. So in that search of enzymes, next slide, ma'am. So generally the limitations of anaerobic fermentation is rate limiting step. So there we have able to see the extra polymeric substrate which is closed, and it needs some cleaving agent to cleave it. So what the microorganism will do, generally it will produce some biochemicals. That biochemicals will cleave the extra polymeric substrates, and it takes some time for doing this process. So we want to minimize that time by using some of the pre-treatment process. So there is different pre-treatment process, physical, chemical, and biological. So we have taken a biological process. Next slide, ma'am. So in this biological treatment process, we can use enzymes and microorganism. When we are using a microorganism, the load of microbe will be again form a sludge. So that should be treated properly, then it should be disposed. So instead of using the microorganism, if we are using an enzyme, it can be reused, and it will not harmful to the environment. So we started thinking how we can use enzymes for pre-treatment. Next slide, ma'am. So we were search of enzymes. So enzymes are generally produced from plants, animals, and different materials, that is majorly from microorganisms and plants and animals. So we want to think how the plants will help in preparing a microorganism, that is enzymes. Then we have found a paper, Thailand scientists have what they have done. So then we have started thinking about garbage and how the garbage is converted into enzymes, and they have given a name is garbage enzyme. But what they did is they have done for 3 months of fermentation, and that fermentation product named as a garbage enzyme, and it contains enzymes, and what are the enzymes it contains was not clearly mentioned. So we have started studying what are the enzymes are there, whether in that particular pH when we are producing a garbage. We can go to the next slide now. Next slide. So this is the overall thing what we have obtained, zero waste concept. So the waste which comes out of residents and the agricultural lands, it will be converted into garbage enzyme, and this garbage enzyme what we have used for industrial sludge degradation, and after this sludge hydrolysis process have been done with the help of garbage enzyme, it enters into that is anaerobic digestion process. There we can able to produce a biogas and some bio solution, and this bio solution can be used as a biopesticides and biochemicals, and then it will be converted into, and if the biogas is used for conversion through hydrogenation process to electricity, and it can be used again to the commercial places and residential places. This is the overall concept what we have taken for our research. Next slide, ma'am. So general production, everyone knows that the one part of I have taken molasses for my study. So 1 part of molasses and 10 parts of water and 3 parts of organic waste. So pre-consumer organic waste we have taken pineapple peels, orange peels, tomato peels, cauliflower peels we have taken. So then we have went for what the procedure is given by Thailand scientists. So 3 months fermentation we did, and then we go for centrifugation. After centrifugation we went for some filtration process to get rid of the cell depreces which is there. Then we have went for environmental characteristics, because we are going to apply in sludge degradation process. So we have checked only the environmental parameters regarding the garbage enzymes. Next slide, ma'am. So these are the parameters what we have checked for garbage enzyme. So when we are checking the pH, it is around 3.6, and total dissolved solid BOD, COD level, and the total protein, and the E. coli CF, that is colony forming units, is less than 3. So it is according to the standard, so we can able to use it for environmental degradation. We have found that. Then next slide, ma'am. And then we have studied whether it is having any antimicrobial activity, because the sludge which have some pathogenic microbes which are commonly found. So we have taken Staphylococcus and fungal Candida albicans, and then we went for fungal solid nutrient agar test and potato dextrose test, and then we have measured the zone of inhibition. Next slide, ma'am. So this is the image what we have obtained, and the inhibition is there with the help of garbage enzyme when we are increasing the concentration of garbage enzyme to 150 microliter. Next slide, ma'am. So this is a table. So in this we have did with the 2 pHs, that is the garbage enzyme pH is 3.6. Then we have adjusted with sodium phosphate buffer to pH 7. At that time we can able to see the zone of inhibition is increasing when compared to 3.6. So this is the study what we have done with bacteria and also with fungal, and the pH 7 is giving a more inhibition rate because of some of the enzymes are become activated during that process of changing the pH. Next slide, ma'am. Next slide, ma'am. Next. This is for fungal, and then we have checked with some biocatalytic activity study. So protease, amylase and lipases assay we have did with garbage enzyme what we have produced. Next slide, ma'am. So we have taken different pH range and we have tested with the proteolytic activity, and we have found around 6.5. The proteolytic activity is high. Next slide, ma'am. And the lipase activity is around 8, and the activity of the lipase is more. Next slide, ma'am. And amylase activity is around 6.5. We have find it is optimal, and the activity is more. When we are adjusting the pH with sodium phosphate buffer, we have found that. Then we have went for what are the organic acids which are found in through HPLC method, and we have taken some of the standards, and with that we have studied. And when the time period of production of garbage enzyme is increasing to 100 days, we have able to find the acetic acid concentration is increasing and all other acids started decreasing, and this is due to the process of anaerobic hydrolysis what happening in the process of anaerobic fermentation which is happening during the process of garbage enzyme production. Next slide, ma'am. So then we have taken individually with the pineapple, tomato, orange, cabbages and mango peels we have taken, and we have produced it, and we have tested the same test we have done. Next slide, ma'am. So in this also we have able to see more or less all the peels are having same phenomena of pH, total suspended solids, BOD, COD, etc. Next slide, ma'am. Then we have, as we have taken this for sledge hydrolysis, whether it is working or not can be identified by some of the characterizations. So we did with the enzyme activity test. So in that enzyme activity like protease, amylase and lipases we have taken. So in that we have found pineapple at the pH of 7 and orange peels with the pH of 7 produces amylase, lipases and protease activity more when compared to when compared to the original pH of garbage enzyme around pH 3.6. Next slide, ma'am. So the same thing as we have taken solid waste, so, that is sledge. So we want to check with the volatile suspended solids reduction is there or not when we are applying a garbage enzyme. So we have tested with the different time period, and we have found at 36 hours we have able to find the major reduction of VS, that is volatile solids, at the pH 7. Next slide, ma'am. So the same thing we have did for total suspended solids also. So in that also we have found at 36 hours the maximum reduction rate was there. Next slide, ma'am. Then we have want to check that soluble chemical oxygen demand. This soluble chemical oxygen demand will tell you about how much conversion, that is hydrolysis, have been taken in the sledge. So when a chain is there, for protein is there, it will be converted into amino acids. When we are seeing a COD, it will tell about how much amount of protein is there, that is organic matter which is present in the sledge. And when we are going for a solubilized COD condition, so in that it will tell how many amount of organic compound which is readily or easily taken by the microorganism which is available in the sledge. So it should be increased, and we have found there is an increase in soluble oxygen demand. So this garbage enzyme is helpful in increasing the hydrolysis rate in this sledge we have found. And the dairy waste sledge only we have taken from the industry for this whole process. Next, and we have checked with the total nitrogen content. Next, ma'am. Total phosphorus content. All these are increasing when we are applying garbage enzyme to the sledge. Next, ma'am. So then we have optimized the production of garbage enzyme using different statistical methods like RSM and artificial neural networks. And finally, we have found come up with a global solution. Next slide, ma'am. Go to the next slide, last. Next. Before slide. So this is the optimal condition what we have taken, and this is the thing what the statistical tools have been given. So we have taken 360 grams of pineapple peels. As the pineapple and citrus peels have given more result, we have taken that too. And then 250 grams of citrus peels. Then we add some, according to the ratio, we have added molasses and water to it, and we have a fermenter, and for monitoring the pH everything we had a setup. So with that we have produced a garbage enzyme, and the pH of the total garbage enzyme, when after adding with the pH change, all these things we have maintained in that reactor. So at the time we have produced protease, amylase and lipases, and we have not went for 3 months of time because I want to degrade only the sledge. So we have stopped the process by 5th or 6th day, and there the optimal protease lipase activity is more during that stage. So we have stopped the fermentation, and that solution is used for the further studies. So then we have went for pre-treatment of dairy waste activated sledge. Next slide, ma'am. So this is a simple concept, that is complex compounds should be converted into simpler form as the microorganism want to take it readily, and it can be converted into other forms like methane or hydrogen, biogas, biohydrogen. So for that solubilization should be done, and we have tested and confirmed the garbage enzyme can able to solubilize the waste what we have taken. Next slide, ma'am. So these are the parameters of dairy waste activated sludge what we have taken. Go to the next slide, ma'am. So these are the effectiveness parameter, how we will find whether the garbage enzyme is effective with this process or not. So these are the things we have increased COD level and SS reduction we have tested, and for this also we have used a statistical tool. Next, go down one. Last time. This is the final thing what we have did. So first we have taken a sludge. After that, we have produced a garbage enzyme in optimal level according to the statistical tool what we have done. Then we have used we have collected the sludge. In that EPS will be there. So the garbage enzyme is helping reducing the EPS complex to some extent. Then we have went for to increase the biogas production rate. We want more degradation process, so we have added with some ultrasound processing along with this, and we have found when we are adding ultrasound process to this, we will get more amount of free amino acids or free fatty acids which can be consumed directly by the microbes, and it helps in producing a biogas. And we have focused on biohydrogen by optimizing the temperature, pH and the environmental parameters. Next slide. So for doing the biogas production, we have ferment, that is we hydrolyzed the sludge and we have kept aside. Then for adding some microbe, that is a seed source for the microbe to do the fermentation process to produce biogas, we have taken a aerobic digested sludge material and we have used as the a seed material or seed microorganism for doing biogasification process. So batch anaerobic digestion we have done. Go down. So these are the two batch we have done with the 250 ml. In that also result came positive. That is injection method. So next one is volume displacement method we have used. So with that we have found we have optimized the conditions also. And after that we have found the production of biohydrogen gas, and it is confirmed with the GC. Next. So biohydrogen confirmation tests have been done with the help of GC, and we have found the biohydrogen is producing from that process. Next one. So it will be confirmed with some parameters of Gumbert's law, and that also we did. Next slide. So in this we have taken three parameters. One is control, where we have not adding any garbage enzyme, treated sludge, and we are not adding any ultrasound. And the second one is only with garbage enzyme, and third one garbage enzyme with the ultrasound, and we have found garbage enzyme with the ultrasound have produced more effect and more biohydrogen when compared to other two. So this is the parameters, and we can able to find the values for the same. Next. So according to the all the results, the garbage enzyme have the capacity to degrade the sludge, that is hydrolysis the sludge, and it can be used in further treatment in anaerobic digestion to produce a biohydrogen process by optimizing the conditions. So these are the works we are doing now. So semi-continuous production of garbage enzyme, and then we are utilizing the sludge which is comes out of garbage enzyme for composting process, and it can be used in agricultural field as a manure. And we are trying to produce a biofertilizers with the nano-sized nano synthesis of fertilizers also we are working on it. Thank you all. Thank you for this.
Thank you so much, sir, for the insightful session. I request Professor Majumdar to please do the concluding lines.
Thank you. I think it's a great pleasure to be a part of this interesting heterogeneous topics. I myself work on a very fundamental area of pure identification and properties of the enzymes. So as you have already listened, Dr. Patanjali talked about layer structure of cultivation. Dr. Arun talked about garbage, how to get the enzymes. Dr. Susan was very interesting because that's something which is closer to my heart. She talked about ALK-B enzyme, which actually we do crystal structure and study. And of course, Dr. Dinesh Mohan talked about making biochar. What I would like to do, I have been given 5 minutes, and I thought that, you know, these experts are here only for a few minutes or 1 hour, and they're coming from different cities, and the audience has a lot of questions. So maybe we could have 1 or 2 questions addressed to individual of these 4 speakers so that we can have the opportunity of discussion. So if there are any question to any of the speakers, please stand up and raise your question. Yeah. Right. Right. I think that's the extremely important issue. Yes, extremely important issue. Yeah. I think you know, at this, these are all, you know, Dr. Mohan mentioned that, you know, minister is interested, and you know, this is something has to be done in multiple dimensions, and the training which he mentioned about, I think I feel that should be spread more, and you can you know, do it in your own place in a way so that biochar can be made, and I'm sure that all other techniques also can be spread in that way. Any other questions or queries directed to any of the speakers?
Actually, I'm very sorry to barge in to answer your question. The Prime Minister has already started, you know, a project for saving Ganga, and there is already a committee in place. Yeah, so I understand. I understand. I understand.
I think the know-how is there, but you know, it needs to be.
Making the point. We well, it starts all from us. Okay.
I think this is an endless discussion.
We have another session. So.
Yeah, you have some point. Yeah.
Okay. The point here, what I'm supposed to mention here, is actually we as conscious individuals, we need to take that step. Now, it's a pressing problem. Since you're in Varanasi, you know the situation well. You understand the situation well. So you need to take that, decision, and you need to start. Probably you might have already done it, or you're doing it. But I'm saying it in general. Whoever is, having that. Okay, we
Yeah,
have
yeah.
learning
I
is a
think
solution.
I think there are issues. Can I just ask her to raise your question? Yeah.
This is the platform to publicize. We have lot many people who are watching on YouTube right now as we speak. So this is the platform to publicize.
I think the message is whatever we learn, we should spread it. I think that's one important thing. And the role of the speaker is there, and those who are participant, they also try to spread it as much as they can. Thank you very much. So with this, I end this session, and thank all the speakers for this session, and thank you very much.
Thank you so much, sir.
Okay.
Indeed, a very good panel discussion we had today. So, I request Professor Majumdar to felicitate our two speakers. Dr. C. Arunji, Dinesh Mohanji, please. Thank you so much. Now I request Shrikantji to felicitate Professor Majumdarji. Thank you so much, everyone. Thank you, all the speakers. Thank you, all the audiences. Thank you so much. Here we come to an end of a very, very wonderful session. Thank you.
I'm sorry we are skipping the tea break. We are doing the last session for the day. It's a panel discussion. Can I please have all the panelists on this stage? Ms. Deepika Jalan. Samira Satija, Madam. Sanjeev Sarangi ji and Shrikant ji. Can I request the presence of Nagesh Vyas, sir? Nagesh Vyas ji.
Congratulations to my soul sisters at the Women's Indian Chamber. Industry Wiki, National Council for Bioenzymes, for being in this Wiki, National Council for Bioenzymes, this wonderful event gathering scientists, innovators, policymakers, for environment, one of the India president. I'm so glad today you will all be coming together to find solutions for nature which are nature-led, technology for ecology, with the help of biotechnologies. Namaste, I'm Dr. Harbeen. My heartiest congratulations to my soul sisters at the Women's Indian Chamber of Commerce and Industry Wiki, National Council for Bioenzymes, for being in this wonderful event gathering scientists, innovators, policymakers, researchers, academics, entrepreneurs in finding solutions for a lifestyle for environment, one of the G20 priorities in the India presidency. I'm so glad to learn that today you will all be coming together to find solutions for nature which are nature-led, technology for ecology, with the help of biotechnologies, biosciences, biosolutions such as the one
in 1940s heralded the era of antibiotics and changed the perspective of the world medicine. Whereas, on the other hand, discovery of bioenzyme is a new dawn that will change the way the world. This life-changing substance made from a simple kitchen waste has the capacity to replace all the heavy-duty cleaners of the face or the surface, which, according to most of us, are presumably the only option that's available in the market today. Bioenzyme applications go a long way, right from healing small skin rashes to eczema, basic pet care to being a remedy to animal diseases like lumpy, water body cleansing to replacing pesticides in agriculture, and much more. Today we have a group of eminent personalities who are not only well researched, but have also executed their findings in various uses of bioenzyme. Let us quickly get introduced to our learned team of experts.
Good evening. This is the last session, I think. This is the last session for the This is the last session for the discussion of entrepreneurs, and I'm calling Mr. Shrikant. Mr. Shrikant is a founder trustee of the BA, and please come on stage and give your words for this. Please come.
Okay. So actually we had 2 sessions planned. One is the enzyme applications and the other one is entrepreneurship opportunities. So since we know that we have not given you a snack break after a long session, a technical session on bioenzymes and on enzymes, and we had scientists over here, so we thought we'll club 2 sessions together. So we are just doing it. So to tell you about the team on the left hand side, we'll be talking about entrepreneurship, but before that we'll have our team members over here. They are young entrepreneurs. They have started the journey with bioenzymes, and they've started exploring and experimenting in various fields. So right from home care to personal care, plant care, planet care, pet care. So there are many aspects that they are working with, and they have been trying their level best, trying to make that mark in the industry as young entrepreneurs. So right now we have starting from let's give gentleman the first preference because he's the only man over here. So we have Bhaskaran from Tanjore. He's an entrepreneur. He's a budding entrepreneur. So he has his organization named Asai Eco Life. And then we have Deepika Jalan. She's also an entrepreneur and also Wiki Bioenzyme Council President of West Bengal. And then we have Yogini Bapat. She is the Vice President of Wiki Bioenzyme Council and also an entrepreneur. And we have Chennai President, Wiki Bioenzyme Council, Vasudha Benjani, also an entrepreneur. She's very famous on Instagram. You'll see her name with Miracle Enzyme is the brand that she calls herself. And we have Ritu Saha, also an entrepreneur. I'm sorry, I just met this wonderful lady. She's not an entrepreneur, but she's here as a chairperson. So she's just going to chair this particular session. And from there we have Ganesh Ram. He's one of the strategists and naturalist, and he's a good friend of ours. And next, I've not met him personally, but he's one of us. So I'm sorry, I need to have his name. Sanjeev. Yes, I have his name. Sorry. I'm so sorry. All right, so next we have Samira ji, also an entrepreneur. Dr. Kritika Jain, who flew all the way from Australia, also an entrepreneur. So they are going to talk about opportunities in entrepreneurship and what are the challenges that we have right now and how do we overcome those challenges and what are the policies that we come to expect from government bodies, or how do we go ahead and establish a strong foothold and ensure that we promote and propagate the concept of enzymes, bioenzymes all over the country. So that's about it. And shall we go ahead and start? All right, so let's start. I'm supposed to be one of the panelists. My name is Shrikant RG, one of the founder trustee members of Bioenzyme Entrepreneurs Academy. So I'll also take the opportunity and with permission from the moderator, Shri Nagesh Vyasji, to go ahead and open the session. All right, so very good evening. So I just prepared a small speech because I don't want to deviate. So today's event is organized by Department of Biotechnology and Academy for administrative careers at the University of Mumbai in association with Society for Innovation in Biosciences and Bioenzymes Entrepreneurs Academy of India. So we bring together a group of eminent experts, leaders from their respective industries and in the field of bioenzyme entrepreneurship to discuss the latest trends, challenges, and opportunities in this exciting and rapidly evolving field. We all know environmental sustainability is one of the most pressing global challenges. But as any emerging field, bioenzyme industry also faces a number of complex and multifaceted challenges from regulatory barriers, technological limitations, to ethical and social considerations. So today's panelists, who represent a diverse range of perspectives and expertise in the field, will share their insights, experiences, and strategies for addressing these challenges and driving innovation and entrepreneurship in the bioenzyme industry. So we have distinguished panel of experts, and we have some questions that we will be asking them. So I take the privilege to ask the questions, and all of the questions. Thank you. So moving on to the home care segment. Do we have Madhumita here? I mean, she's also one of the panelists. We don't see her here. Yeah. Thank you. Okay, so she's Madhumita. She's one of the entrepreneurs from Coimbatore. She has a brand by name Enzyme. So I have prepared my questions to ask Madhumita also, please. So that's the reason why I remembered somebody is missing. Why are 2 chairs empty?